and Jim already introduced me. I work at the California Water Science Center, and I'm actually not exactly sure how I ended up giving this talk. I think Jim saw a couple of USGS talks and said, hey, we should get USGS to do a session, and he suggested that I just stand up and kind of tell you what we do at the California Water Science Center that you might be interested in, both in bioassessments and general ecology. So the topics I'm going to cover, I thought I'd actually point out a couple of things that we have at the Water Science Center that you may or may not be aware of. A lot of you work with pesticides and water quality in general, so I thought I'd talk briefly about our pesticide lab and some really cool stuff that's being done with monitoring water quality in our center. And then I figured I'd give you kind of a brief survey of a lot of the studies we've done in the past and kind of what we have ongoing right now, just so you can see kind of the range of stuff that my group, which is the Aquatic Ecology group, have done and are doing now. And so this talk is actually pretty data-free, which is kind of unusual for me, but it's kind of a retrospective and current status of ecology at our center. So, not sure what's going on here.
Might try going back there.
Yeah, go back.
Okay. Right. Okay, so just a couple of caveats. There are actually many other USGS people doing ecology, aquatic ecology, and other kinds of ecology in California. One being the National Research Program, which Terry Short, who's coming up next, is part of that. And the Western Ecological Research Center, which you may be aware of, they do aquatic stuff, but they also do a lot of terrestrial and avian ecology and plants and a variety of other stuff. And I'm not even covering everything we do at the California Water Science Center that is at least connected to ecology, like the hydrology and sediment groups. So our center has a lot of stuff going on at any particular time. I was gonna time myself. So the first thing I wanted to mention was the Pesticide Fate Research Group, which is actually an internal lab at our center. They actually work a lot with the national lab that Pete mentioned in methods development. It's actually a pretty active group and pretty large. And their major focus is kind of the fate of current use pesticides, and so what kind of happens to them in the aquatic environment. So they're applied, they get into the water, so they're interested in where they go after that, whether it ends up in biota or water that people use for whatever reasons. And they tend to focus on kind of program-specific or study-specific kind of stuff, so they can do really focused type of methods development and studies. They really look for very low method detection limits, so they can see really small quantities using small sample volumes. So I think they're actually like analyzing individual bees, which is the thing at the top. I'm not sure why they're analyzing Skittles, but I think that's for the pharmaceutical slide. And they do collaborate a lot with toxicologists and ecologists around the state, and I know they've done work for various water quality studies in California. They're really, their methods development is really driven by the California kind of pesticide environment. We have so many different ag products being produced, and there's constantly being new products brought on the market for specific purposes, for different crops, and also as various things are found to be bad for the environment. As you know, new stuff comes online to replace those, and over the life of this graph, it's an average of 11 new pesticides used in the Central Valley Basin each year. So it's a really challenging environment to do chemistry in. I'm not going to go over this. They've developed a lot of methods. They continue to improve them. They can find almost anything at parts per trillion levels, if there's stuff in particular that you're interested in. They spend a lot of time on methods development. And this is just a list of some of the recent studies they've done. I think the native bee one got a little press recently, but neonicotinoids and pyrethroids and disinfection byproduct stuff they've all done studies on all those. They've also been collaborating on the Delta Regional Monitoring Program results, a graph there. And they're finding kind of the typical thing we seem to find with pesticides in California streams. They're really complex mixtures of a lot of pesticides that individually don't exceed criteria, but together probably are having bad effects on aquatic ecology. So that's the lab. The other thing I wanted to mention was this high-speed mapping of water quality that Brian Bergamaschi's group has developed. I added the title in quotes, which is water quality at 20 miles an hour. You basically outfit a boat and you drive around and you collect water quality as you go. They basically run their boat as they would going between place to place and take a ton of data. This is something they developed because a lot of the projects they had started working on, were getting away from kind of sampling water quality at one particular place over time, like on a weekly or monthly schedule. They're very familiar with the latest technology that is coming on the market. So they were seeing that they were now getting sensors that were good enough and fast enough to take a lot of data in a really short period of time. So basically they took this whole list of instruments shown on the slide, stuffed it into a couple of suitcases, connected it to a GPS system and a data logging system. So you connect all this up to a laptop on your boat, you have a water pickup on the bottom of the hull, and you drive around. They've mainly been using this in the Delta, and this is just an example of some of the tracks they've collected. You can compare between seasons there. So this is just showing nitrate and chlorophyll because there's a lot of interest in primary production in the Delta right now, as this seems to be a food-limited system for a lot of the aquatic organisms. And then you can look at individual tracks in more detail if you want to. The colors are actually on a different scale in each one of these, so you can't compare colors between, but you can see here there's some actual concentrations added to look at. So if you combine this kind of spatial density with a subset of fixed sites in the Delta, you get a really good picture of both spatial and temporal variability in water quality. So it's something we're just starting to look at as far as connecting it up to fish and zooplankton production, but I think it's going to be a very powerful tool in the very near future. So it's really cool. So that brings us to my own group. It's mainly been myself, Jason May, and Marissa Wolf for a number of years. We actually brought Fred Feirer on board 2 years ago now. He's like many young people, young scientists brought in. He has a lot of energy, a lot of ambition. So he's actually been bringing a lot of new projects into the group, and he's actually, I think, he might even be getting up to 6 or 8 staff. So he's really developing a strong program focused on the Delta for now, but he's starting to move into other areas as well. So it's really nice getting some new energy into the group. So this is just kind of a long list of stuff we've done in the past. Pete mentioned the Aqua Program starting in 1991. That's how I got hired at USGS. I got hired to work on the San Joaquin River Drainage Study Unit. And California actually had 3 of these study units, so went from the San Joaquin to the Sacramento to the Santa Ana. I stopped putting dates on it after that because I was feeling old. Actually, Terry Short, who's up next, also started, I think, almost exactly the same time, within like about a month. So NAQUA actually brought a lot of aquatic ecology into what we call now the water discipline of USGS. So in the blue are kind of the ongoing stuff. In white is kind of in the past. This is just kind of a list of what have been my favorite projects over the years. So National Water Quality Assessment Program. We did a lot of work in Whiskeytown in kind of the early 2000s. Experimental watersheds, and I'll show you some slides of some of these. And our ongoing stuff is Santa Ana River native fishes and the stream quality assessment, which actually Jason has been spending a lot of time helping get that up, especially the site selection. So we've had a lot of experience in a wide range of habitats, ranging from the headwaters of the lower San Joaquin River. This was very disappointing. I think Terry may have forgiven me by this time for taking him down the Salt Slough and Spanish Grant Drain. I'm not sure. Santa Ana was actually a very interesting project because you got to see Santa Ana varies from a very urban environment to actually a very nice environment, kind of in the upper headwaters. So on the left is kind of the gradient for the Santa Ana, and on the right are a number of the pristine tributary streams flowing into it at various points. But it's actually a very productive system, just very few species. Kings River was nice. I'd never actually worked on real first order and second order streams before, so this was a great opportunity. We did some work on diatoms up here, found them to be very sensitive indicators of stream environment. Each of those slides actually does have a stream in it. They're just so small, you can't see them. I mean, you can literally walk across these streams, but a very beautiful setting. And then Whiskeytown, we actually got, I think we worked there, you probably worked there for 5 years. I worked there for about 3. Doing a variety of stuff, bioassessments. Jason did metals work, and then we actually did a little fire ecology stuff. I really liked that project because we got to work with really cool species, Pacific giant salamander, tailed frogs, and then native rainbow trout and sculpins. The fire, this was actually, I forget when I think the fire was in 2006. It was actually really hot in spots, but it was kind of heterogeneous, so you'd have a hot spot and then an area that didn't burn that much. This is 2 slides in actually pretty much the same place. So you see the grass that was seeded to prevent erosion. I was actually really surprised. These streams pretty much recovered from this fire in like a year. You went back the next year and pretty much the bugs and the fish all looked the same. So now we're working down in the Santa Ana. There actually still are native species there. The map is, where the Earth view is our study, study area. So the headwaters of this part of the Santa Ana are actually up here at the Rialto wastewater treatment plant, then flows down, goes into the Santa Ana River channel, which is actually dry above here for many miles. A major tributary is the Ricks wastewater treatment plant. And then the area with the most of these 2 species in it is kind of above this. Actually, you see Riverside Drive is right there. So this is kind of the hangout of these 2 native species in this area. The Santa Ana sucker is a federally threatened species, so there's a lot of interest requirement to protect it. So we were asked down mainly because the agencies down there are developing a habitat conservation plan, which kind of gets all their regulatory requirements in one package. As it turns out, there'd been quite a bit of work on the sucker, but they still didn't really have a good idea of how many there were. So there hadn't been a good population estimate or what habitats they were using. So we're actually down there looking at those things right now. So it's kind of a really cool project for me anyway, because I'm actually get to go out in the field and snorkel in wastewater treatment effluent, and it's really quite fun because it's kind of back to basic fish ecology, which is what I've been trained in. We've also had a lot of office projects, modeling and analysis. These slides are actually a training class that Ian Waite organized. I think Jason and I are actually in there, but you can't recognize me because my beard is still brown. And this is kind of the point where we started collaborating with a lot of the people in the room. We started off doing a lot of modeling actually with Ian, starting off with the western states where NAQUA studies had been done. Then we did a focused one on Southern California and then moved on to the Northeast. We actually participated in kind of a national synthesis of studies of urban streams that had been done around the country as part of NAQUA. Recently, we've actually been collaborating a lot with SWRCB and Department of Fish and Wildlife. We've had 2 recent projects looking at habitat flow biology modeling, and I think Rafi is going to talk a little bit about one of those projects. And an ongoing project is with the Nature Conservancy, and actually Darren Carlisle, who's actually on Pete's team, where we're actually modeling natural flow characteristics of California streams, working with Jeanette Howard and Julie Zimmerman from the Nature Conservancy, and Jason's going to be talking a little bit more about that project. Okay, I'm going to go through this really quickly. So this is some of the stuff actually that Fred Fierer's doing. You can see he has a lot of studies going on. He's a very busy guy right now. So he has a project looking at Rodeo Lagoon, which is actually just outside of San Francisco Bay in the Golden Gate National Recreation Area. Mainly interested in effects of eutrophication on the tidewater goby population that lives in the lagoon, because that's a protected species. Some actually really cool work with Sacramento Splittail, which is a native minnow. They've noticed a high incidence of scoliosis, which is the bent backbone, which is kind of a trademark of selenium toxicity. And they're looking at deposition of selenium in the otolith, the ear bones of the fish. And they've actually determined that this is something that the young splittail are picking up in the environment. If you look at that lower left graph, you see the ring. The other pathway would be in the egg from the mother, but that ring indicates they're picking it up as they start feeding as very young fish, which actually has a lot of implications for where they're picking up the selenium and where regulators need to look at the sources. Working with smelt, probably the notable thing is the lower left panel showing the smelt cam, which is actually a camera which you put on your net, so you actually see the fish going through the net instead of having to bring them on the boat, which when you're dealing with a rare endangered species is a big step forward in sampling. Longfin smelt is a California endangered species. He's doing work with that. Kind of the biggest thing we have going on is we're starting to look at tidal wetland function in a big study being funded by the Bureau of Reclamation. mainly working in the North Delta and the Cache Creek area, if you're familiar with that. And I'm actually working on climate change issues where we're taking climate change outputs, using what we know about how fish use different parts of the environment, and then projecting how much habitat will be available. Here this is just kind of a validation exercise using 2011 and 2014. Starting in the next couple of months, we're going to actually start getting data from climate projections out the next 100 years and starting to do this kind of thing with different climate change projections. So that's going to be very interesting. So I don't know, I probably don't have time for questions, but no, I don't.